Hello and welcome to an episode of Momento Malum today. For this episode, we'll talk about the use of genetic markers in apple breeding. We'll start off by covering the nature of genetic markers, followed by the use of genetic markers in terms of discovering the genetics of flavor, and finally some genetic markers covering the nature of disease resistance, specifically fire blight resistance. Let's get started. Many agronomically important traits such as yield, quality, and some forms of disease resistance are controlled by many genes and are known as quantitative traits. Within each crop, there are specific regions of the genome, and within these regions of the genome, there are various genes that are associated with the traits of interest. This location is called a QTL. One of the chief breakthroughs in the nature of how QTLs operate is that there are regions in the genome that genes connected to a specific trait are connected to in a phenomenon called linkage. This means that if a region is inherited, at least a portion of the genes that are associated with that trait will come along for the ride. Genetic markers are and continue to be made to find and detect the presence of such regions. This allows for easier selection for specific traits and the furthering of the knowledge of genomics. Throughout the decades, various genetic marker technologies have been made. These include AFLPs, SNPs, and many others, all in the service of detecting such regions, and each marker technology having their own limitations. One of the most common methods in modern times, one of the common methods of detecting QTLs will be SNP genetic markers. And SNP genetic markers will be the key item for the next two topics we'll cover. One example of the use and search for genetic markers is in the paper regarding the aromatic compounds of apples. A portion of the apple's flavor is determined by its aroma, or how it smells. The reason behind this is beyond the scope of this video, but a great many of the aroma compounds are esters. Such aroma compounds include but are not limited to pentyl acetate, which has a banana scent, hexyl acetate, which has a pear-like scent, ethyl-2-methylbutanoate, which is strawberry-like in nature, and butyl acetate, which has a fruity flavor that's found in many apples and several candies. The apple cultivar with the highest concentration of butyl acetate measured is Ducat. This cultivar comes from Kazakhstan and is primarily eaten fresh or dried. This is a hybrid between Dr. Oldenburg and Cox Orange Pippin. Dr. Zoe Mikhailovsky and Mr. Taib Sumru did extensive research into the genetics of flavor using the method GWAS, a particular methodology that will be covered in greater detail in a future video. What you need to know is that through this method, the non-coding region in the apple's second chromosome called CHR21164-704 was discovered. This region was found to be associated with the above esters mentioned. Theoretically, in the future, using this marker, one could screen for improved flavor in apples based on this knowledge. We will conclude by talking about disease resistance. Fire blight is one of the most devastating diseases in apples. This bacterial disease causes the full death of the tree, unlike most other common apple diseases, which mostly cause fruit damage. Over the course of many years of study, Dr. Awaz Khan obtained a great deal of understanding regarding the regions of fire blight resistance within the apple genome. One QTL and its associated SNP, LG07, is found in Cox Orange Pippin and is associated with improvements in fire blight resistance. The LG12 region is found in the crabapple cultivar Everest and is likewise associated with improvements in fire blight resistance. Screening different cultivars and seedlings for these markers would make it easier to improve fire blight resistance within apples and crabapples, potentially increasing the longevity of an apple orchard. And that about covers everything. Thank you for watching. Thank you for watching this video. If you enjoyed this video, please like and subscribe. If you really enjoyed it and think you can donate, you can do so at Buy Me a Coffee, link in the description below. Thank you for watching.